So I'm from University of Colorado, the Renewable and Sustainable Energy Institute that we formed very recently, and also Liquid Crystal Materials Research Center, as well as the Department of Physics. In my talk, I will start from an introduction to liquid crystals, to different types of phases that we could have in the liquid crystal state of matter, although I will not be able to overview even a small fraction of them because there is a huge variety of liquid crystal phases, and it would be impossible to do it in even 4 hours. Then I'll show the basics behind the work of liquid crystal displays and a lot of electro-optic devices that we use in our research and also in different types of consumer devices in our everyday life, and in particular displays. In the second part of the lecture, I'll then tell you how you can also use light to switch liquid crystals, to realign optical axis of liquid crystal using tightly focused laser beams, and then we'll discuss possible applications and uses of this. So let me start from a very brief introduction to liquid crystals. You already heard the lectures by Jiuchang Li and also by Professor Takezoe during the first week of iCamp in Sydney. So you already had a little bit of exposure to liquid crystals. I'll give you a bit more of introduction because this will be useful from the standpoint of view of understanding how liquid crystal materials can be switched optically. Right? So you all know about crystals and you all know about fluids. You know the distinct characteristics of those states of matter. So crystals are solid materials. The molecules or atoms are not allowed to move with respect to each other. And in crystals you also have crystalline orderings of you have both translational and orientational ordering at the same time. In isotropic fluids, the molecules are allowed to move with respect to each other. That's why the fluids can flow. Liquid crystals stand in between. They combine the properties of crystals and fluids in a very unique way because, in a sense, you take advantage of both. Those materials can flow just like liquids, so they can be easily processed for many different applications, but at the same time, they do possess ordering just like crystals. And what's interesting about liquid crystals is that they can combine those properties of ordering and fluidity in a variety of different ways. And that's why we have so many different types of phases that I'll show you some of them, most typical, most commonly used and most commonly encountered in several minutes. Well, but how do we actually characterize liquid crystals? Well, I mentioned that they have some degree of order, although maybe less order than crystals. They have some partial order, right? But at the same time, they can also flow. Also, ordering of liquid crystals is usually characterized by the liquid crystal director, and this is the average local orientation of molecules of liquid crystals, right? So you can imagine liquid crystal being composed of some cylindrical molecules that slightly misalign with respect to each other, but on average are all pointing in the same direction. Right? And this direction we call liquid crystal director. It's director because you cannot distinguish the head and tail in here, right? Because the liquid crystal phases such as nematic phase have non-polar symmetry. Now the other characteristic that's very important for liquid crystals is the orientational order parameter, which we'll denote as S in here. And this is nothing else but second Legendre polynomial. What it lets us to do is to characterize how strongly those lipid crystal molecules misalign with respect to each other while staying locally ordered and locally oriented on average along the same direction, the director, right? And so the maximum value of S is 1. That's when we would have a crystal, right? But minimum corresponds to isotropic state. Then that's when we would have no order at all, right? And so when you take an average of second Legendre polynomial, this gives you information on how where theta in here is the angle between those molecules and the director. The value of S gives you information on how much ordering you have in the liquid crystal state. And so in here you can see temperature dependence for the most common liquid crystal phase, the so-called nematic liquid crystal phase depicted here on the left. Right? So you can see that close to, as a function of temperature, the value of order parameter can change, and at lower temperatures it's higher. At very low temperatures it could approach unity. Usually it's smaller than that. At room temperature of many liquid crystals it could be, say, 0.7, 0.6. And then when you would heat your liquid crystal material further, at some moment you would have discontinuous transition to the isotropic state, and that's when the order parameter value discontinuously changes to 0, right? Because in isotropic fluids, there is no order parameter is equal to 0. Well, there are so many liquid crystal phases that we want to subdivide them to different groups. And two different classes of liquid crystals that we often distinguish are the so-called Lyotropic liquid crystals and Smalyukh liquid crystals. And you probably can guess, even from the name, that in Smalyukh liquid crystals, the liquid crystal state is observed somewhere in the temperature range between the isotropic state at higher temperature and crystal state at lower temperature, right? So in those materials, the liquid crystal behavior is observed as you change temperature. Well, in Lyotropic liquid crystals, you usually have a composite system of some kind when you mix different materials, usually very different materials, and those could be surfactant molecules, such as the soaps that we use to wash our hands in the morning, with water, right? And so you might know that the surfactant molecules, such as the one depicted in here, have hydrophobic tails, those hydrocarbon tails in here, and hydrophilic heads over here. Right? So when you mix such molecules with water, then what happens is they self-assemble into different structures so that the hydrophobic tails are hidden from water while hydrophilic heads are exposed to water, right? And so one type of such a structure is bilayer of those molecules, and we will see later on there are many others still. And so usually the shape of molecule or building blocks such as Micelles or other building blocks predefines the types of phases that we can observe, right? And so you can understand this because we can have molecules of different shapes. For example, we can have rod-like molecules, right? And for physicists, imagining that a molecule like this has a rod-like shape already means a lot, because we can understand the interactions and phase behavior just knowing only this, right? And then you can also have disc-like molecules. You could have also molecules of many other different shapes. The only requirement is that those are strongly anisotropic molecules, right? And then you can expect liquid crystal phase behavior, right? Either while you will be changing temperature or as you will be dissolving those molecules in other solvents, and you can observe phase behavior, liquid crystal phases. So the most common type of phase is the nematic phase. In this state of matter, we have only orientational ordering and no positional ordering at all. Right? So in here you can see two different types of nematic phases formed by cylindrical molecules, like those here, and also by disc-shaped molecules shown in here. Right? And those are the chemical structures of typical molecules that can form those phases. Well, so what you see is that the cylindrical molecules in here simply align parallel, on average parallel with respect to each other, while they are able to move with respect to each other, and that's why the material flows, so there is no positional ordering. In the case of the disc-shaped molecules, simply the normals to those coins align in the same direction, which would be vertical with respect to the screen, and again, you have orientational ordering only. Well, you could also have a variety of different chiral phases, and again, the most common one I show in here, where the molecules, the cylindrical molecules of liquid crystal twist with respect to each other along this helical axis chi in here, and the distance over which they twist by 360 degrees is called cholesteric pitch. Right? So the molecules that will form this type of phase are very similar to the ones that form nematic liquid crystal phases, and usually the difference is very minor. All we need is one carbon chiral center that will break the symmetry. You will now lack the mirror symmetry of the molecule, and those will then self-assemble into cholesteric liquid crystal phase. And what's beautiful is that you can vary, you can tune this distance that we call cholesteric pitch over a very broad range, from something like 50 nanometers to hundreds of micrometers. And you can do it either by using different chiral molecules or simply by mixing molecules that have this chiral center with the molecules that don't, right? And so then you use a simple relationship that relates the cholesteric pitch with the concentration of this chiral molecule in the mixture of the two, of chiral and non-chiral molecules. Right? So we can form self-assembled cholesteric phases of this kind. And the reason why it's so simple, why they assemble, is because, as I mentioned it already, the molecules of liquid crystal in this case lack the mirror symmetry. Right? So our hands, for example, are chiral, so you wouldn't be able to superimpose a mirror image of it, of the hand with itself. And likewise, when you add the chiral center in here, and we are trying to show a three-dimensional picture in this way, then the mirror image is not superimposable with the molecule itself. So the molecules, while assembling into the nematic phase, forms this twist, and this gives rise to the cholesteric phase. Well, there are many other phases, and so in the nematic and cholesteric phases, we had only orientational ordering, but no positional ordering at all. So that means centers of mass of those molecules were completely disordered, just like in water, right? But now you can imagine that the molecules can form layers, right? And so in one direction, in this case, those molecules will look like solid because there is some positional ordering. So as you would go in this vertical direction, you will see the modulation of density, periodic modulation. But within the layers, there is no positional ordering at all, so that the molecules are free to move with respect to each other. Right? So those materials can be sort of being two-dimensional fluids, but one-dimensional crystals. Right? And apparently they can have a huge variety of different phases. For example, the so-called Smectic A phase, where the molecules are perpendicular to the layers, or the so-called Smectic C phase, where the molecules are at some tilt angle with respect to the normal to the layers, and many, many other phases. Well, apparently so many that you can write a book on just the variety of phases that can have one-dimensional positional ordering combined with two-dimensional fluidity. Now, just to give you an insight on what other distinct type of phases could be formed, from the optical standpoint, we could have uniaxial liquid crystals or biaxial liquid crystals, right? So why liquid crystals are so interesting is because they are, from optical standpoint, uniaxial, biaxial crystals with different symmetries, but they also respond to a variety of external factors such as electric field and laser beams very easily, and so that's why they are very interesting materials. But what you see in here is the biaxial smectic liquid crystal phase, where you can see that molecules have some plank-like shapes, and they do form the layer structure. The long axis of those molecules on average point perpendicular to the layer planes, but the shorter axis of the plank-like molecules also have some ordering within the layers. This is only orientational ordering, right? So from the standpoint of positional ordering, those are still one-dimensional crystals, two-dimensional fluids, but in addition to the orientational ordering along this axis, you also have orientational ordering within the planes of the layers, right? And then you can also have biaxial nematic liquid crystal, where there is no positional ordering at all, right? But the difference from uniaxial nematic liquid crystal is that those are now plank-like molecules, and they order themselves into the structure where there is orientational ordering along the long axis, and also the short axis orientational self-order in this direction too, right? So you would now need two directors rather than one, and those are biaxial crystals. And then there are a variety of other phases: ferroelectric, anti-ferroelectric. For example, the one I show in here has the molecules tilting with respect to the layer normal, but the tilt direction is alternating from layer to layer, and those have anti-ferroelectric properties, while the material I'm showing in here is the so-called Smectic C star, and in this phase you have a combination of layer structure and chirality, because the molecules are tilted with respect to each other, but this tilt direction is changing as you would move in the direction along the layer normal, right? And so it's changing in a continuous way, the structure is chiral again. So the molecules that form those structures again would lack the mirror symmetry, but because they form the Smectic C-like phase, the combination of tilt in the layers and also the chirality gives rise to the ferroelectric properties, which you can imagine are very interesting from the standpoint of many practical applications. Then there are a bunch of different phases that can combine the presence of defects in the ground state with all the features of ordering that we have discussed, right? And so one classic example I'm showing in here, imagine you have Smectic A-like phase that we discussed before, but because you would have some molecules that also have tendency to form chiral structures, having lacking mirror, because of the lacking of mirror symmetry, and so those materials can form the so-called twist grain boundary phase, where you have the blocks of smectic material separated with respect to each other by the boundaries of formed by these screw dislocations, right? And so it's a fairly complicated phase, right? We'll not go into details, but it's also all of this self-assembly is happening on the nanometric scales, and it's very interesting from fundamental standpoint, but also from the standpoint of applications. Now, I described the cholesteric phase before. You can also obtain a three-dimensional twisted phase, the so-called cholesteric blue phase, and apparently there are at least 3 different varieties of those. So usually in the blue phase, molecules also would like to twist with respect to each other, but they do it differently than in cholesteric phase. So the basic building block in here is the so-called double twist cylinder. So imagine your cylindrical molecules which lack mirror symmetry, so they have some chiral centers. They would point along the cylinder axis in the center, but then as you would move away in the radial directions, they will twist in all radial directions, right? And so this is the double twist cylinder. There's a benefit of doing it this way from the standpoint of molecular chirality, and then those double twist cylinders can assemble into the cubic phases, such as body-centered or face-centered cubic phases, or also amorphous phases, where, such as blue phase 3, in which case you have a spaghetti formed by those double twist cylinders, right? So spaghetti-like structure. And so what's interesting again is that the periodicity of those phases can be on the scales of hundreds of nanometers. This makes them very attractive from the standpoint of photonic applications, because they can be essentially self-assembled photonic crystals. And although not with the full band gap because of relatively low refractive index contrast. The materials like that can be used as, for example, self-assembled lasers when you add dyes to them and pump some light in, you could see such applications in the work done in this direction in the lecture of Professor Takezoa from Tokyo Institute of Technology during the first week, right? So many different phases, but all of those that I described so far were formed by small molecules, and small molecules is something on the order of a nanometer, right? So relatively small organic molecules. Now imagine that you have those molecules linked with respect to each other, and forming polymeric chains, right? So when the mesogenic units of polymers have strongly anisotropic shape, rod-like or disc-like, you again expect the liquid crystal phase behavior. And so in here you can see the side chain and main chain liquid crystal polymers, right, that again form a variety of liquid crystal phases. And so essentially all the phases we discussed and even more can be formed by polymers. So in this case, the only difference is that the molecules are linked with respect to each other because they are connected to the polymer backbone somehow, right? And so again, a huge variety of phases, a huge variety of different possibilities for applications. Now, you can also have other types of building blocks. Not necessarily those have to be individual molecules. For example, you can form liquid crystal phases out of micelles of different shape, and those could be either spherical micelles or cylindrical micelles. You can form liquid crystal phases out of bilayers and a variety of other building blocks. So for example, in the case of the micelles, cylindrical micelles, you can form hexagonal phase and you can form a nematic phase. You can form a lamellar smectic-like phase out of bilayers. And if you have a surfactant, then as a function of concentration, you can observe all of those phases, right? Because at very low concentrations of surfactant in water, you would observe some small number of micelles that form above some critical micelle concentration. Then as you will keep increasing concentration, a variety of phases will form, and depending on temperature and concentration, there is a very rich phase diagram of those phases, right? And what's beautiful is that this ordering is occurring by itself. You don't have to nanofabricate those details, those features on the scales of nanometers, right? The other type of building blocks is shown in here. So what we have is stacks of dye-like molecules. I'm sure that many of you had molecules similar to this one for your breakfast today, right? Because many food colorants are forming liquid crystal phases, right? When you dissolve them in water. For example, there is sunset yellow, that's very typical food colorant, and it forms a phase like this, right? A nematic phase, a columnar phase, depending on concentration. And so what happens in this case is that you produce stacks of those molecules. They self-assemble, right? You don't have to do anything, just mix the two. The molecules like this and water, for example. And in this particular case, because you have the hydrophilic water-soluble molecules on the periphery, those charged groups over here, and an aromatic hydrophobic core inside, the molecules tend to assemble into the long stacks so that they can expose the hydrophilic parts of molecules to water, right here on the periphery of those stacks, while hiding the hydrophobic parts of the molecule from water, right? And so now, as you will form those long stacks of molecules, you can form either nematic phase. So in this case, the stacks have orientational ordering but no positional ordering, or you can also form columnar hexagonal phase and many other phases. Many nanoparticles, such as carbon nanotubes, such as different types of inorganic nanorods, can form liquid crystal phases too, and in here I just give some examples, right? So all you really need is to stabilize those nanocolloids or colloidal particles, and as long as they have shapes anisotropic enough, they can form liquid crystal phases too. This is an example of the carbon nanotubes forming liquid crystal phase when they are stabilized using strong acids, dissolved in strong acids, and the example in here shows carbon selenide nanorods in a similar way forming liquid crystal phase. Well, still we are far from even briefly overviewing the liquid crystal phases, right? So what I show in here are liquid crystal elastomers or liquid crystalline rubbers. We use rubbers all the time, right? We know what kind of properties they have, very useful. That's why we use them so often. But the liquid crystal rubbers can have even more interesting properties, because in this case the orientation of molecules is coupled to the mechanical properties or to the shapes of the whatever you produce out of such an elastomer, right? And so the types of phases that we overviewed before, nematic, smectic, columnar, they all can be formed by elastomers, so liquid crystal rubbers too. And so what happens in those materials is that the molecules are relatively restricted with respect to moving, from the standpoint of moving with respect to each other, but they can rotate, right? And so depending on the phase and depending on the fields that you might apply, the orientation will change, and this will then couple to the mechanical properties, so to the shape, for example. And if you will on purpose change orientation of those molecules in the rubbers, you can utilize such materials for applications such as artificial muscles, for example. And so as you would also go from the isotropic state of such a liquid crystal elastomer to a nematic state, you observe spontaneous change of the shape, and you can also induce those changes optical and electrical, and this is why they are very interesting. And so I'll not dwell on mechanical properties of those, but I mentioned already, very interesting, because realignment of molecules in those rubbers gives you another degree of flexibility, another degree of control, and the possibility of controlling those mechanical properties, optical and electrical. Well, liquid crystals are a big part of biological materials too, right? So many biological molecules such as lipids, actin, microtubules, proteins, viruses, even bacteria are known to form liquid crystal phases, and that includes molecules such as DNA, the carrier of genetic information. And why do they do it? Well, if you just look at molecules like this, like actin or DNA, you realize they have strongly anisotropic shape, right? And so as you would increase concentration of such molecules in a solvent like water, then what happens is at some critical concentration, you spontaneously transition from a disordered state to the liquid crystalline ordered state. And the theory by Onsager for the case of rigid rods precisely gives you the value of concentration that you need in order for this to happen, right? So this value of concentration is something of the order of 4 ratios of diameter to the length of such a molecule, right? And so as soon as you go beyond this critical concentration, you observe spontaneous formation of liquid crystalline ordered state. Well, of course, the molecules in biology and particles that we might use are often more complicated than just rigid rods of this kind. So the molecules of DNA, for example, are flexible, and so you still can account the flexibility of those semi-flexible biopolymers in finding the critical concentration for the spontaneous formation of the liquid crystal state, and there are those series available. But again, as I mentioned before, the very most important biological molecule of DNA, as you would keep increasing concentration, does form a liquid crystal state of matter, and it can be nematic, cholesteric, or columnar liquid crystal phase. Now, in here you can see an example of the ordered states, liquid crystal states formed by DNA molecules, but similar ones can be also formed, for example, by FD virus, which is nothing else but a rod that has a diameter of about 6.6 nanometers and the length of about 880 nanometers. Right? So it's a semi-rigid rod where you have RNA in the core and then some protein coat. That's it, basically, right? But from the physics standpoint, it behaves as a semi-rigid rod, and at high concentration, again, it forms the liquid crystal phase. And then you also observe the liquid crystalline phase for phases in many biological and zoological instances, right? For example, in the spider silk, you can observe liquid crystal phase, a liquid crystal self-assembly, and in many other examples that are not overviewed because of the lack of the time. So how does a liquid crystal sample look like as you would observe it in a microscope? Well, if you would put a liquid crystal sample between 2 cross polarizers and then observe between in the microscope, what you observe is something what you see in here, right? And so this is provided that your liquid crystal sample is not aligned in some ways, right? And so the reason you have this variation of intensity transmitted between through the sample between cross polarizers is because the liquid crystal is optically anisotropic material. I mentioned already that from optic standpoint, liquid crystal is a uniaxial or biaxial crystal. And so as light would propagate through this liquid crystal slab in here, it will change polarization, right? And because of that, the light can leak in here, passing through the polarizer, the liquid crystal sample, and then analyzer. And apparently this is a way we usually image liquid crystals to understand how they align in the sample. Because unlike in the case of solid crystals that many of you are familiar with, when we have a cube of crystal with uniform orientation of the axis, in the case of liquid crystal, if you just put a drop of liquid crystal on a substrate, the optical axis will not be uniform. It will change with coordinates, right? And so we often need to be able to control how it changes with coordinates, and we also need to be able to image those changes. And so this is the simplest way of doing it. But of course, this type of imaging provides only two-dimensional picture of what might be happening in the three-dimensional sample, because the light is passing through the entire liquid crystal slab and gives you only two-dimensional resolution of the orientational ordering in such a liquid crystal sample. So you apparently can calculate the intensity as a function of orientation of director, right? So the angle that the optical axis makes with respect to the polarizer in such a cross-polarizers scenario. However, if you know the orientation of optical axis, it's easy to calculate what pattern of intensity you will have in such an image. But it's more difficult to do a reverse analysis and be able to deduce the orientation of optical axis from an image like this, right? Because you have an integral over the depth of such a sample, and so that means intrinsically you have only two-dimensional resolution. And so a better way of imaging is to use a confocal microscopy that is extensively used nowadays in biology, for example. So I assume that most of you know that what you do in confocal microscopy is you use a laser to excite some dye molecules in the sample, and then the fluorescent light from those excited molecules can be detected, for example, using the very same objective in the microscope, and then you have a pinhole in here right in front of the detector that allows you to block the light that would go from places in the samples away from the focal plane, right? And so because of that, you obtain resolution along the optical axis of the microscope too, and that means you get the three-dimensional resolution. So it can be diffraction limited, and with good objectives can be on the order of a couple hundred nanometers. But in most biological applications, what you are interested in is how different molecules or parts of the biological tissue are spatially distributed with respect to each other, right? So what you focus on is on labeling of those different molecules or tissue with different dyes, and then you can visualize what's happening in your sample. What we are interested in is how orientation of molecules changes in the sample as a function of coordinates, right? And what you could actually have is 100% the same one type of molecule in the entire sample. What you are interested in, how orientation of those molecules changes in the sample. And so now, in order to be able to visualize orientation of those molecules, you usually dope such a sample, such a liquid crystal sample, with the dye molecules that also have rod-like anisometric shapes, and therefore they align with the liquid crystal molecules, so that when you will use polarized excitation, the intensity of detected fluorescent signal will depend on orientation of those molecules, and this will be the way for you to visualize three-dimensional patterns of orientation.
You should break for 10 minutes if you're in a starting point, or
yeah. Well, so let me maybe keep on for like 2 more minutes, and then sorry. I guess you got excited. Okay, so okay, so in here is an example showing how this type of imaging can be done for a liquid crystal cell, similar to what we use in the displays, right? In the displays, we apply electric field to a liquid crystal cell like this and realign the liquid crystal molecules. Well, it's hard to know how those changes happen across the sample thickness or across the display thickness, and a confocal microscope, like we just saw, will allow you to visualize those changes. And so the changes of intensity of light here depicted using the colors allows us to visualize those, the orientational distribution of the molecules in the liquid crystal sample. The reason you are able to do this is because the strongest fluorescence intensity corresponds to the case of polarizer being parallel to the transition dipoles of the dimolecules, and the weakest to the case when they are orthogonal. While in the situations in between, there is a simpler relationship that you can utilize to reconstruct the patterns of molecular order. So maybe we'll stop in here and resume after the break.